Yeah, thank you very much, Enrico, and thanks a lot for Mitocon for all they do to support this project. I'm very grateful for that. So this is a long project started several years ago, but what changed now is finally is being accepted, so it should be online soon, finally. And then also we have the clinical trial starting this year, so that's why they asked me to put more emphasis on this part, which I will do at the end. But first, let me start by starting what is our approach. Our approach is we're trying to find ways to accelerate drug discovery, which we know is very challenging for rare diseases, and we know how lengthy the process can be. And so one possibility is also to, instead of using de novo drug discovery, to use drug repositioning. This is also what we do to try to accelerate some steps. Of course, with that comes also some challenges because the market might be small for rare diseases, and also the fact that these repurposable drugs bring the challenge that there might be some less enthusiasm from pharma, and we can discuss at the end if needed. And the other aspect that we're working on is, of course, this new alternative method, NAMs, and in our case, brain organoids, which might have some advantages, for example, in finding individual brain areas or looking, for example, a specific more aspect related to development. But of course, there's also some challenges, for example, in the fact that they're not mini brains. They have a lot of things that they're missing in terms of vasculature or immune aspects, and also they're disconnected from the environment and the rest of the body, so it's not a picture of what is really happening in the brain. Despite that, what we're trying to do is to use these NAMs and also repositioning of drugs to see if we can accelerate some aspect of identifying treatments for mitochondrial diseases, and in particular, Leigh syndrome. I will not mention the details of Leigh syndrome because we already heard about that. I just want to mention that our focus is mainly on variants in the gene MT-ATP6 and SURF1, which are among the most common ones. And this is the one where we have generated the iPS cell models and brain organoids. And so a few years ago, when we looked at brain organoids, we noticed that so here in red, these are neurons. You see in the SURF1 mutant ones, there's a defect in neurons, but not only. We see also defect in how progenitors here, how they're organized in the control and how they're not in the mutant. So this also came to the fact, to our understanding that at least in these models, probably because they are disconnected, they are under more stress and so on, we see something accelerated that wouldn't happen in the brain later on. And so we think there is already some progenitor or organization that is disrupted. So that came to our concept that if this could be something that we could use for screening as a platform. And so we thought if this process of going from stem cell to neurons is disrupted when mitochondrial disease mutations are there, so maybe we could find something that we could already use neuroprogenitor cells, which start to use OXPHOS as a model. And also, so we have then two ways that we did screening. One is to look at neural outgrowth, so that you look at how neurons are able to regrow, and then the progenitor's function. So the first one, we use it in collaboration with computational scientist in Luxembourg, Antonio Del Sol, because we, instead of making a large screening, we thought maybe we could accelerate it further, but first in making some deep learning screening to identify some hits that then we could then validate. So that's what we did, and basically we looked first at the single-cell RNA sequencing of brain organoids, and based on the trajectory of progenitors, radial glia, and neurons, we identify some aspects that can be improved, or some genes whose expression should be there in order to allow this this transition in a physiological manner. And so then, Antonio, they used these differentially expressed genes to identify something that we could modify with drugs, and they suggested some drugs that we could then test, and that's what we did. And this came then to the fact that we found two azole compounds, or mostly one talarazole, that was improving brain organoids formation, but also other aspects. And we think that these are going through the retinoic acid mechanism. That could also explain why this could be important for neurogenesis, but at the same time we identify also aspects on cholesterol metabolism and also bioenergetics. This is just a summary, but I'm telling you because this is an example that we think, oh, maybe talarazole could be an interesting drug, but then what next? What next is that is a bit depressing, is because although there has been study in mice using talarazole as for osteoarthritis, in humans, there was a clinical trial for talarazole in adult with osteoarthritis that is ongoing or something, but we tried to discuss with the pharma to obtain some of these drugs. It was not possible, and probably they decided to discontinue. Probably, I assume this trial maybe is not successful, but the bottom line is that we don't have, this drug is not made because it was originally made for psoriasis, but there's other drugs, so it is potentially safe, but we need to find a way now to make it, produce it, so we have to see. So this is just to say that maybe we have enough, maybe we accelerate, maybe we do drug repositioning, but then maybe we come to that, which is then cannot be necessarily accelerating because we're not, there with the drug yet. So, but then this comes to the next part, which was the title of my talk, where we instead used a real screening, and this was on neuroprogenitor cells. And this was because we identified some years ago that neuroprogenitor cells with mutation in the MT-ATP6 genes have increased hyperpolarized mitochondrial membrane potential that we could, and because of that we noticed some dysfunction in calcium, and we could use this MMP as a screening target. So together with Ole Pless in Hamburg, we developed this screening, and we made several lines of ATP6 and controls, because we cannot do CRISPR-Cas to correct the mutation because it's a mitochondrial gene. So we had to have a lot of control and mutant lines. And to make the story short, after this screening of repurposable drugs, we identified some PD5 inhibitors that could depolarize the abnormally hyperpolarized mitochondrial potential without causing loss of viability. And with the PD5 inhibitor, we prioritize sildenafil because it's safe for pulmonary hypertension in children. So we started looking at the mechanism of action, and with Manuela Bottani in Verona, we looked, there is some improvement in the ATP amount, but the bioenergetic effect overall was not too strong. So we see, for example, complex 5 activity is a bit improvement, not significant. MyTDNA copy number, a bit improved, not significant. So we don't see a stronger bioenergetic amelioration. But the mitochondrial potential that it was high in a lot of different lines, it is ameliorated. But then we thought maybe it's just uncoupling, but then that would not be good, because if we have a lot of other mitochondrial DNA defects cause mitochondrial potential decrease, so then we don't want it to further decrease. So we measure in SURF1 mutant lines where MMP is decreased, but then with sildenafil was increased. So we don't think that then acts as an uncoupler, but what we think is what I mentioned before about calcium. We saw an increase in calcium, and I will go in a few minutes to that point. In parallel, Stefan Jacobs and in Göttingen found that when we look at MPC's structure of MIC60, they think that there's some abnormality. You see some black here in the middle, then when it's treated it's not, so there might be something that can help there in the crystal formation. But this is the picture still not clear. So then we thought, okay, we did more analysis, more omics, and of course more brain organoids. And this defect that I mentioned before, that we see in the Leigh syndrome organoids of a sort of progenitor disorganization, we saw it also with ATP6, and it was not corrected fully with sildenafil. But there were some correction in some specific parts and some related to differentiation, and in particular at the level of progenitors, we identify PRKG1, which was dysregulated and then increased with the treatment. So PRKG1 is a cyclic GMP-dependent protein kinase 1, is downstream of cyclic GMP. So the way we interpret that is after with sildenafil, when PD5 is inhibited, then there's more cyclic GMP, and that could lead to PRKG1 increase or activation, which is known that this can in turn have an effect on neuronal outgrowth or calcium. And so that's why in our mind, the calcium homeostasis regulation comes before the MMP modulation. We did some tests in brain organoids where we sliced them and then we apply acute metabolic stress. And we see then when we do that, the ATP6 organoids have increased calcium release, but the one that were pre-treated with sildenafil they were not showing this. And then the pretreatment in neurons increased the neuronal outgrowth, which would fit with this sort of idea that now we have. Although, as you can see, this is as you can know, this is a drug that probably have a lot of polypharmacology action. So I don't think that we have. This is the only way that might be doing something, because of course, like I mentioned here, we don't have vasculature, so all the effect on endothelial we didn't even look at. But we think this PRKG1 might play a role because when we inhibit, with siRNA, we knock down PRKG1 in control neurons, we see a defect in neuronal outgrowth. So we already hear from Dario Brunetti before the data in piglets, but first we also collaborated with Dao Fudai, Johns Hopkins with the induced knockout mice, where they found that sildenafil was increasing a little bit survival of these mice, and also the energy expenditure, and also the PRKG1 again expression in the brain. And these are the data from Dario that you've already seen. And I want to also highlight here the fact that also PRKG1 there was increased, although not in all the treatment animals, but in some of them significantly. So, of course, the last part is the most exciting because we know that it can be given to patients and to children. So Markus Schulke started individual-based treatments. And he looked at the Newcastle scale, and this is the natural history study from Berlin and the one from Newcastle. And every point is the same patient monitored at different time points, and you can see that basically the severity goes over time, always increasing. And in the 6 patients that they were treated, instead, after treatment, this was going no more always as a severity. Of course, in this patient, there was a further up, but there was first a dip down. So overall, seemed promising to suggest there might be delaying some disease progression. So with that, we were able to obtain this orphan drug designation. And now, thanks to Sympathetic Consortium, we are now planning the trial. So originally, when we discussed with Emma about the trial, we thought we could do multiple N01 trials. So, okay, the challenge, of course, is a rare, a rare population, so we need to collect. So in order to collect less patients, but then they didn't like it because we proposed multiple crossover, and so We now proposed a single crossover so that we have 2 time points, first sildenafil and the other placebo, and then 1 crossover. We should get the feedback now in a couple of weeks. That we submitted late last year the optimized plan, and this is the plan that we're doing is as a primary outcome to use the Newcastle scale. We are looking only at MT-ATP6 variants, and mostly with a Leigh-like phenotype. So that means that we're looking at variants with high heteroplasmic levels, so NARP patient would not be part. Also because sildenafil might have a side effect on retinitis, and so that we could also be present in NARP. So that's what we pick as a safety. And also after looking at the data in pigs about the high doses, and also the data from the STARS-1 trial that was in pulmonary hypertension, we don't use high doses. There will be only low and middle doses. So the number that has been calculated is now 50 in total to reach a statistical based on this Newcastle primary outcome. And the sites so far will be in Nijmegen, the Netherlands, and Berlin. But we are discussing additional sites in Italy and France. And so this is, they might be able to join later on once we started or as soon as the paperwork is ready. So with that I conclude. So I told you about the talarazole and the challenge that now we have ahead. I told you about sildenafil and how we hope that we could, by using this, going from patient and go back to patients, hopefully to find something that can have some positive outcome on the patient. And thank you very much for your attention.